Good morning, boys. Welcome to the online classes once again, class 12. I hope you had a wonderful, restful Dasai and restful vacation. Now let's begin with the next section. What we have discussed so far is the different type of genes that are present, structural and whatever, different type of jumping genes, transposons and all those. Now we'll talk about linkage. Now, before we understand what is linkage, it is very important that we need to remember that is, there are far more number of genes than the number of chromosomes. As we know, we have only 23 pairs, but the number of genes is in thousands. That is why a single chromosome will carry a number of genes together. Each chromosome will carry a number of genes together. Each chromosome may carry more than 100 different types of genes. And as we know, you take for example, human genome is between 25,000 to 30,000 different type of genes that is present in the 23 pairs of chromosomes that we have. So this 25,000 to 30,000 number of genes is present on the 23 pairs of chromosomes. So what happens here is the closeness of the genes. Some of the genes are tightly placed, or we can say they are closely placed to each other, whereas some genes are at a distance. Those genes which are located on the same chromosome tend to transfer itself during gamete formation together. They have a tendency to be transferred together during the gamete formation. This particular phenomenon, the tendency of genes to be transferred together during gamete formation, is what is known as linkage. Now, there were a number of dihybrid crosses conducted by Morgan, like Mendel's dihybrid crosses, but there is a difference. When we talk about Mendel's dihybrid crosses, that is law of independent assortment, the ratio, the result in the F2 generation is 9:3:3:1. That is what we have already studied. Because Mendel was very lucky, the genes did not cross over, they were closely linked with each other. So, they have a tendency to remain together. But when T.H. Morgan, another scientist, conducted the same experiment of the dihybrid crosses, and he had selected one of the most used genetic sample, that is Drosophila melanogaster. Drosophila has few number of chromosomes, and the pattern of transmission is more or less the same as that of humans. In the case of human beings, we have XX is the female and XY is the male. Same in the case of Drosophila also, XX is the female and XY is the male. That is why T.H. Morgan studied the sex-linked genes. That means those genes that were present on the X chromosome. And he conducted a dihybrid crosses, but the result were very different from that of Mendel. Mendel's dihybrid crosses ratio is 9:3:3:1, but here it was completely different. Morgan selected two Drosophila. He hybridized these two Drosophilas, one with yellow body and white eye. You already know dihybrid means there are two genes that are present. Yellow body with white eye. We take two genes into consideration. And another he took was brown body with red eye. He selected this two, and out of this, these two are both are parental generation. The red eye, brown body and red eye is called the wild type. Now we need to understand what does the wild type mean. Wild type basically means dominant. It is slightly different from the way that we represented in the Mendel's experiments. We wrote capital letters for dominant and small letters for recessive. Here we represent them slightly different. Remember, wild type means dominant and this is the recessive. So brown body is the wild type, so that is dominant. We represent this like this. The yellow body with white eye, we represent it with small yellow and small w. Small yellow and small w. This is the recessive type. So what we got to remember here is, this is the recessive type. Now, how is the dominant type or the wild type represented? We take the same alphabets, but here we put a cross mark, a plus sign. This plus sign indicates that they are wild type or the dominant type. So when Morgan conducted this experiment on Drosophila melanogaster using these two varieties, yellow body and white eye, and red eye with brown body, in the F1 generation, they did not segregate. Segregate independently of each other in the F2 generation. They did not segregate independently like in the case of Mendel. That is why the ratio was different from 9:3 to 3:1, which was the ratio of Mendel. The parental generation were more than non-parental generation. In the F1 generation, entire thing was parental generation. In the F2 generation, the parental generation was more than the recombinant generation, or we can say the non-parental generation. So basically the linkage means the genes that are present on the same chromosome have the tendency to be transmitted together in the next generation. During gamete formation, they do not separate. This is basically what is linkage. Now we move on to the types of linkage. Types of linkage. In this we will be talking about first complete linkage. In complete linkage, what happens? The genes are very close to each other. So they do not separate from each other during gamete formation. In complete linkage, the entire gene that is present on the chromosome is transmitted together. They do not separate or segregate even during the gamete formation. This type of linkage is said to be complete linkage, and we do not find any recombinant or any crossover or any new variety. We will see only the parental type that is present. They are transmitted as a unit. Take for example, we take a condition, heterogamete and homogamete. We take a cross of heterogamete and homogamete. Heterogamete will obviously have two different, and homogamete, both the gene chromosomes will carry the same genes and at the same loci. So during gamete formation in the F1 generation, The type of gamete that is formed here is of two types, two different types, A and B and A and B. But here this is homogamete, so it produces only one type of gamete. When this crosses with this, the result is now in the F1 generation, what we see here is both the gametes are, when they combine with each other, both produces the parental type. So what we see here is 100% parental type. This is what is said to be a complete linkage. But whereas when Morgan studied Drosophila melanogaster, the majority of the offspring in the F2 generation were dominant or the wild. What we see here is 100% parental type. Now let's move on with Morgan's dihybrid crosses. Morgan selected two different types of Drosophila. Drosophila melanogaster is a very important material for genetic studies. The reason is it carries very few chromosomes, and number of chromosomes and number of genes are present on the chromosomes. And another reason why we select Drosophila melanogaster is the sex determination. Like in our case, XS is female and XY is male. And same in the case of Drosophila also, XX is female and XY is male. That is why we take Drosophila as a very important genetic material. Morgan selected two different varieties. One with yellow body and white eye, and another brown body, red eye. So what we see here is we are taking two different characteristics into account, the body color and the eye color. So this is a dihybrid cross. Now we need to remember here, yellow, like in the case of Mendel, the dominant were written with capital letters and the recessive were written as small letters. But here in the Morgan's crosses, the dominant are written with a plus sign. We'll talk about that. Yellow body and white eye. Yellow body and white eye. Yellow body and white eye is represented by small y and small w. So from this we need to understand this is a recessive type. Now how do we depict red eye, which is the wild type? Now here you need to remember that wild type means the dominant type. The dominant is the wild type. This is represented as Y plus and W plus. The plus sign indicates that this is the dominant and the wild type. So what Morgan did was cross these two different varieties of Drosophila. He took two different parents. He took yellow and white, yellow and white female. This is the female, and brown and red as the male. So what we get here is, this is homogametic, so we'll get only one type of gamete. Only one type of gamete. This is the parental generation. Parental generation. The gametes that we get here is only one type because this is homogametic, and here we get two different types. Two different type of gametes like this. Now the Y chromosome does not have the reciprocal of this, so that is why we have one and the other is the Y chromosome. When this combines with this is the result that we get. Wild type female, wild type female, and the recessive type male. Recessive type male. This is in the F1 generation. This is in the F1 generation. We see the parental type, both the parental type that we see here in the F1 generation. Now when the F1 generation is selfed, this F1 generation is selfed, it produces 4 different type of gametes. This produces 4 different type of gametes. 2 are the parental type and 2 are the recombinant type after crossing over. Let's draw that one as well. What we get here is 2 different type of gametes. 4 different type of gametes, sorry. One, two, three, and 4. We get 4 different type of gametes here. 4 different type of gametes. Whereas in the case of male, we get 2 different type of gametes. The 2 different type of gametes will be, this is in the case of female. In the case of male, we'll have 2 different type of gametes. And this. Now, when this is crossed in the, when it is selfed in the F2 generation that is produced, the F2 generation that is produced is when this combines with this. When this combines with this, now when this combines with this, we get this one. When this combines with this, These are the female population that we get. 2 are the parental type and 2 are recombinant type. Similarly, in the case of when Y chromosome combines with this, we'll get parental type. When Y combines with this, we'll get another parental type. And when Y combines with this, and when Y combines with this, we get a recombinant type. Now, when actual field study was done, it was found that 98% was of parental type. Now, when the field study is done, not looking at this ratio, if you look at this ratio only, then you will get confused. When the field study was done, 98% was parental type and only 1.3% was recombinant type. So from this what we deduce is the genes are close to each other and they are completely linked. Though there we see a very minute percentage of crossing over that has taken place, but the majority, 98% is of non-recombinant type. So from this what we deduce is this shows a complete linkage. So this linkage does not correspond to the Mendel's law of independent assortment, that is 9:3:1. The ratio that we normally see in the Mendel's crosses, 9:3:3:1, we do not see that. Because here it is complete linkage. Now, you can cross this with this, you can cross with this with this, this with this, this with this. These two produces the parental type and these two produces the recombinant type. But if you only look at this, then you will get confused. Remember, the field study was done and in that 98% was parental type. Okay, so this is complete linkage. Now let's move on to the next type of linkage that we come across is incomplete linkage. Now when we talk about linkage, we already understand that these genes which are present on the same chromosome tends to transfer together during gamete formation. But when the genes are located at a distance, it is not necessary that they are transmitted together. So here crossing over takes place, and due to crossing over, the genes are separated. And this is the case when the genes are located at a distance from each other. This is called incomplete linkage. To understand this, we'll again look into Morgan's experiment. Morgan selected two different varieties of Drosophila, white-eye with miniature wings, which is the recessive type, and red-eye with long wings is the wild type. White-eye with miniature wings. Now, this is just to give you an idea that these genes are located at a distance. White eye and miniature wings. And wild type, that is the dominant type male that we are taking here, is the red eye with long wings. Red eye with long wings. As we have already talked about, the corresponding genes are not present on the Y chromosome. That is why we leave the Y chromosome blank. Like in the first case, we can produce the same type of crosses. Here, the gamete that is produced again is of one type. white-eyed with miniature wings. And here we again have, this is the female and this is the male. Here we again have two different type of gametes. And the Y chromosome. When this is crossed with this, again in the F1 generation, we get wild type with long wings, because this is dominant. Wild type with long wings. And when this combines with this, we get white-eyed with miniature wings. This is red-eyed with long wings, and this is white-eyed with short or miniature wings. Similarly, now when this F1 generation is sieved, we again produce four different type of gametes here. Here crossing over cannot take place because these are heterozygous. So when crossing over takes place here, it will again produce four different varieties. Two will be of parental type and two will be of crossover type. I will just write down the gametes only. The gametes that is produced here is two of parental type and two of crossover type. Two of crossover type. Obviously over here we will have two gametes because they are heterogametic. The result that we get after the cross in the F2 generation, we get to understand that 62% is of parental type in the F1 generation, but here what we see is 37% is of recombinant type. From this we deduce that when the genes are located at a distance from each other, there is every chance of them being separated and forming a recombinant type. Now, there is a very important point that we need to deduce here. As far as the crosses are concerned, you try to do it yourself. If you cannot, then you can always ask me in the WhatsApp. I will again draw it and explain it to you. But remember that plus sign means wild type and wild type is dominant. Now, the most important thing that we can deduce from what we have discussed here is there is linkage. That means every gene has the tendency to be transmitted together from one generation to another. But when they are close together, they are not separated. But when they are at a distance, they can be separated. And when they are separated, it shows incomplete linkage. Recombination is a measure of distance between the genes located on the same chromosome. And this knowledge is also used to create genetic maps. The human genetic map, the genome study. In that also, this linkage is a very important, you can say, study which helps us to map the genes on the chromosome.